In this section, we will talk about cytogenetics. So let's talk first about basic definitions and concepts, and then we'll talk about aberrations of chromosome number. We'll talk about euploid abnormalities, as well as aneuploidies like trisomy and monosomy. And then we'll discuss aberrations of chromosome structure, translocations, deletions, inversions, and isochromosomes. So let's discuss a few definitions here and talk about what a chromosome is. Remember that in the phases of the cell cycle, in G1, in S, and in G2, DNA is loose in the cell nucleus and has no distinct structure. It's just kind of flaccid and looks like a bowl full of spaghetti, right? Now in the M phase, the chromosomes condense to form the structure that you see here, the structure that we classically call a chromosome. So remember, in a mitotic chromosome, we see two sister chromatids held together by a centromere. Again, telomeres referring to the ends of the chromosome, and then the chromosome being further classified by short arm and long arm, depending on where the centromere is, short arm being called P and long arm being called Q. Now the two sides of the chromosome are identical, the result of DNA replication, so they're called sister chromatids, right, and are connected by a centromere. Chromosomes can be described based on the location of their centromere. Different types of vocabulary, a couple more terms here to learn. Metacentric is if the centromere is in the middle, acrocentric if it's towards one side, and telocentric if it's all the way to the end. Now another vocabulary term, karyotype. If we take all of these chromosomes that are in mitotic phase and we order them, we pair them up into the chromosome pairs, the homologous pairs, then we see what we call a karyotype. And here we have a classic example of karyotype. We see chromosomes 1 through 22, the homologous pairs, and then we see a pair of X's. So this would be a karyotype for a female. Now, euploidy and aneuploidy. Cells are euploid if they contain a number of chromosomes equal to a multiple of 23. For example, haploid if they're 23, like we would see in a gamete. Diploid, 46, which we would see in most cells of the body. Triploid would be 69, or tetraploid would be 92. Those are all euploid cells. Aneuploidy is the term used if cells do not contain a multiple of 23 chromosomes. For example, in Down syndrome patients, there are 47 chromosomes with an extra 21, so those cells would be considered aneuploid. Now, how does aneuploidy arise? Remember, going back to meiosis. Remember, we have this diploid progenitor cell. That diploid progenitor cell will replicate its chromosomes, right? So that we see there in that second step, each chromosome has two sister chromatids, so it's a homologous pair. Remember, during meiosis 1, the homologous pairs separate, right? So you get genetic content reduction right at that step. Now, during meiosis 2, we get separation of the sister chromatids in each of those daughter cells. So at the end, this is a mitosis-like division, so that at the end of meiosis 2, we have 4 haploid gametes, each with half of the DNA content of the original diploid progenitor cell. Half of them will have chromosomes of maternal origin, half of them will have chromosomes from paternal origin, and of course the chromosomes are mixed and matched. There's not one cell that gets chromosome 1 through 22 from the mom and then from the dad. They mix and match, they independently assort, right? So where do we get aneuploidy at? Two different parts. We can either get it at meiosis 1 or meiosis 2. If we look at non-disjunction at meiosis 1, that means that the homologous chromosomes don't separate, right? Non-disjunction. So gametes or children are missing a chromosome or have two copies from non-identical grandparents. What does that mean? Look at the diagram. So during meiosis 1, we get failure of those homologous chromosomes to separate. So on one side, we get both homologous chromosomes, and on the other side, we don't get any chromosomes, or empty cells, right? Now, going through meiosis 2, those homologous chromosomes will each separate by sister chromatids, right? So they'll line up along the mitotic plate, just like in mitosis, and each of the daughter cells during meiosis 2 will get one sister chromatid of maternal origin and one sister chromatid from paternal origin. Now, at the end, we have each of those having extra amounts of DNA, and then on the other side, again, going through those blank cells or empty cells, they'd have a lack of DNA. Now, this same kind of thing can occur during meiosis II. Let's say meiosis I happens like it's supposed to, and we have separation of the homologous pairs. Now, if you see on the left, we get separation of the sister chromatids like they're supposed to separate, with each daughter cell getting one sister chromatid. On the right-hand side, we have a non-disjunction at meiosis II, and both sister chromatids go into the same daughter cell. So it actually ends up having more DNA than it's supposed to, and then again, the other daughter cell would be an empty cell since those sister chromatids fail to separate, and it would have no genetic material. So what are the consequences of a meiotic disjunction? They can be monosomy, right? We have a normal gamete that's haploid, could fuse with an empty gamete that lacks a certain chromosome or genetic content, and when those two fuse, we have a monosomic embryo. Instead of having two chromosome 21s, for example, like they're supposed to, it only has one. That would be a monosomy at chromosome 21. On the flip side, if we take a normal gamete that's haploid and fuse it to a diploid gamete, one that had, for example, a non-disjunction during meiosis II, which is what we see here, two sister chromatids, they were supposed to separate and they didn't. So when we get fusion, we end up there with, instead of two chromosome 21s, for example, we would have three chromosome 21s, and that's where we get trisomies, right? Trisomy, that would be trisomy 21. So, if that is trisomy 21, very common, Down syndrome, let's look at a few of these characteristics. Most common autosomal trisomy, 1 in 800 live births result in a trisomic chromosome 21. Features that you see here, mental retardation of varying degrees, IQs anywhere from 20 to 70, short stature, the characteristic facial features that you see here in the picture, that depressed nasal bridge, the upslanting palpebral fissures, and the epicanthic folds. Now, what you may not be able to see in the picture, but are also very common in Down syndrome, are things that happen inside. Congenital heart defects, very common in trisomy 21. Early onset dementia, and also increased risk of leukemia. Now, Down syndrome is associated with advanced maternal age due to maternal meiotic non-disjunction. 80% of these, it's been found, these non-disjunction events occur at meiosis I and 20% at meiosis II. As far as paternal age goes, there's been no correlation with advancing paternal age and Down syndrome. 4% of cases of Down syndrome are familial, usually due to a Robertsonian translocation, which we'll talk about a little later. 1% of cases are due to somatic mosaicism, meaning a mitotic non-disjunction. These are individuals that started out as normal zygotes and then during the process of mitosis had a non-disjunction event. We would call that somatic mosaicism. Looking at a typical karyotype here, we see at the 21 chromosome section down there, big red circle to mark it out, 3 chromosome 21s, so that's a karyotype for Down syndrome. Now, Edwards syndrome, another trisomy. This is trisomy 18, approximately 1 in 8,000 live births, so a lot less common than Down syndrome. Severe mental retardation, rocker bottom feet, low set ears, microagnathia, or small jaw, congenital heart disease, flexion of fingers, which we can see in the picture there, prominent occiput, and death usually within the first year. The keys that you want to go into the boards for this one, the rocker bottom feet, the microagnathia, and that finger flexion, those are commonly used in the boards to try to get you to think about Edwards syndrome. Patau syndrome is another trisomy, trisomy 13, approximately 1 in 6,000 live births, so about as common as Edwards, not quite as common as Down syndrome. Again, severe mental retardation, characterized by microphthalmia, microcephaly, cleft lip and palate, polydactyly, congenital heart disease, just like the other two trisomies, and death within the first year. The giveaways here, cleft lip and palate, most common in Patau syndrome, and polydactyly. Sex chromosome aneuploidies can also occur. One of them is Turner syndrome. This is actually a monosomy. This is the only viable monosomy that we see. It's 45X, so they're lacking one of those sex chromosomes, marked by short stature, ovarian dysgenesis, primary amenorrhea, and then physically you see a webbed neck and commonly cystic hygromas. Klinefelter syndrome is a trisomy with XXY genotype marked by tall stature, long arms and legs, testicular atrophy, and gynecomastia and female hair distribution. So here we've got another opportunity to practice our pedigrees here. Look at this one and tell me what you think. We've got all generations being affected. We've got males and females both inheriting. Any specific inheritance patterns that you see here? In that first to second generation, we see the male passing it to both females and males. And in the second to third generation, we see mom passing it to son, and we also see father passing it to son. So taking all of this into account, it looks like it would be autosomal dominant. Another question for you, Tay-Sachs disease, is it autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive, or mitochondrial? The answer here is autosomal recessive. Remember, this is one of those storage diseases. Those loss of function type of enzyme diseases, always look for autosomal recessive. All right, let's continue on with our cytogenetics, and we'll talk about translocations. Translocations are exchanges of genetic material on one chromosome with genetic material on another chromosome. Two types of translocations that we want to know about: reciprocal translocations and Robertsonian translocations. With reciprocal translocations, what happens here is we get a swap between two chromosomes that we call balanced. This means that there's no net loss of genetic material. So we can see in the diagram here we have the blue chromosome and the orange chromosome, and they have genetic material that just trades places. But if you look, we still have all of the genetic material for the blue and all of the genetic material for the orange. There was nothing lost. So we would call this a balanced reciprocal translocation. Now, offspring of individuals with a balanced translocation are at risk for developing a partial trisomy or partial monosomy as a result of abnormal pairing of the derivative chromosomes during meiosis. What does this mean? So if you look here, what we have is that we have that reciprocal translocation that happened in two of the chromosomes. So when these homologous pairs go to line up during metaphase 1 of meiosis 1, remember they line up according to homologous DNA. They look for similar DNA. But since we have that translocation, what happens is the orange maternal chromosome wants to line up with part of the orange from one paternal and part of the orange on the other paternal chromosome. So we get this X-type formation. The blue chromosome will do the same thing. So what happens here is we get this type of tetraploid chromosome pairing. Now, when those chromosomes break up, we could get partial aneuploidies depending on how they decide to segregate. If we get alternate segregation, where we get a whole blue and a whole orange, or we get those reciprocal, those balanced translocated chromosomes where we have no loss of genetic material, then we're fine. We get balanced gametes. Now, if we get segregation that's unbalanced, what would happen is we get one blue yanking another part of the blue, or one of the balanced translocants, but with a normal chromosome. So we get the normal blue and then a balanced blue-orange. And then the other possibility would be the normal orange with, again, that translocated blue-orange hybrid. We see that if we get either one of these cases, we'll end up with a loss of material. In that first case, we'd end up with a loss of orange, and in that second case, we'd end up with a loss of some of the blue. So this is how we end up with partial trisomies or partial monosomies as a result of abnormal pairing. Now, the other type of translocation we see is a Robertsonian translocation. And this is a fusion of two acrocentric chromosomes. Remember, acrocentric are those that have centromeres that are towards one side. Now, what happens when these chromosomes fuse, we get loss of the p-arms of both of those chromosomes. Usually, this has no phenotypic consequences because those p-arms are so short. What we see here, diagramming, looking at the diagram, we have the blue and the orange, both have very short p-arms. If both of them lose those p-arms and fuse together, we call that a Robertsonian translocation. So now we have this hybrid blue-orange with no usual phenotypic consequence. Now, offspring of a parent with a Robertsonian translocation can develop partial monosomies or trisomies like we saw with the reciprocal translocations, right? So what we see, depending on how the pairing goes, we can see that hybrid chromosome pairing with one orange and one blue, which would give us normal offspring, right? We wouldn't lose any genetic material. If that hybrid chromosome pairs with an orange and then you get another orange coming in, then we can get that partial trisomy, or what can happen is you can get the blue coming in, we can get the partial trisomy the other way. Lots of these mixed and matched combinations which will result in trisomies or monosomies, again because of that extra genetic material that the blue chromosome doesn't know that it has, that happens to be the rest of the orange chromosome. Now, familial Down syndrome is a good example of a consequence of Robertsonian translocations. So what we have here is we get this Robertsonian translocation between chromosome 13 and chromosome 21. Okay, so let's say we have the mother that has this Robertsonian translocation. It's got this hybrid chromosome, and then it's got a normal 21, and she's got a normal 13. The father also has two normal 21s, two normal 13s coming together. Now remember, during meiosis, they're going to contribute certain genetic material, only half of their genetic material. Let's say that the mother contributes her normal 21, but instead of contributing her normal 13, she contributes the hybrid 13. And let's say the father contributes his normal 21 and a normal 13, which is what he has. Now that offspring is going to have normal 13, but he's going to have trisomy 21, extra 21, because of that Robertsonian translocation onto the 13 from the mother. Inversions is another possibility. What we have with inversion is portions of a chromosome that become inverted by aberrant crossover events during meiosis. So inversions basically means a piece of the chromosome in crossing over will flip. And you can have either pericentric or paracentric inversions. And all that means is if it involves the centromere or not. So you see on the left, we have a pericentric inversion where the top part of that chromosome gets flipped around, and since it involves the centromere, we call it pericentric. On the right-hand side, we have a paracentric version where it's just Q arms, and when the Q arms translocate, they flip, and so that instead of having the order yellow, orange, purple, black, you have the order black, purple, orange, yellow. That's an inversion. Duplications mean that portions of a chromosome become duplicated, just like the name suggests. For example, in the case of an isochromosome, here we have an isochromosome, which basically means here, looking at the diagram, what you have is that Q arm of the X chromosome becomes duplicated and it actually takes over the P arm. The P arm becomes lost and you have a fusion of two Q arms and you have what's called the isochromosome XQ. This is the only isochromosome that is compatible with life. So if you see isochromosomes or XQ, they're talking about this duplicative phenomenon, and this is the only one that is compatible with life. Now, deletions are loss of parts of chromosomes, as the name suggests. This can cause disease by loss of essential genes. A few common diseases that you want to be aware of that are caused by deletions: Cri-du-Chat syndrome, which is a deletion in 5P of the 5P arm, Angelman syndrome and Prader-Willi syndrome, which are both deletions of the Q arm or parts of the Q arm of chromosome 15. So let's take a look at Cri-du-Chat syndrome. Again, this is deletion in the 5P arm. Very rare disorder, from 1 out of 25,000 to 1 out of 50,000 people, characterized by microcephaly and severe mental retardation. Also characterized by a high-pitched crying or mewing, which is where the name comes from. This is French for cry of the cat, so Cri-du-Chat. Result of this deletion. Cardiac abnormalities are common in these kids, and the children generally do not survive to adulthood. So for this disease, what you want to look at is the P arm on 5, and you're looking for a deletion up in that telomere. It's kind of hard to see on the karyotype, but if you look at the diagram, you can see that the telomeric portion of the P arm there, 15.2 and 15.3, have been deleted. Now, Angelman and Prader-Willi syndromes. 46 deletion of the 15q 11 through 13 loci. Exact same deletion resulting in two very different diseases. You can see from the photos here, Angelman syndrome on the left, they get severe mental retardation, they have ataxia and seizures, and they refer to this as the happy puppet syndrome because of these atasic movements that they have. Now, Prader-Willi syndrome, on the other hand, moderate mental retardation, not as severe as you see in Angelman syndrome, also marked by hypogonadism and obesity. So, that begs the question. Angelman and Prader-Willi syndromes have very different phenotypes, but result from the same deletion of chromosome 15. If the affected chromosome 15 is of paternal origin, the individual develops Prader-Willi features. If the affected chromosome 15 is of maternal origin, the individual develops Angelman features. So here we have, specific to paternal versus maternal, what type of genes are going to be expressed and what type of disease you're going to develop. This is called gene imprinting. When we talk about paternal versus maternal differences, we talk about imprinting. So these syndromes, depending on the parental origin, again referred to as imprinting, is a function of different gene expression at a locus related to parent of origin specific DNA methylation patterns. Remember that you, as you develop, will methylate DNA in certain spots, and it just so happens that paternal and maternal methylation will be different. So depending on the parent of origin DNA will determine the type of disease that will result from the deletion. If we look at this diagrammatically, it becomes a little bit easier to understand. So here we have chromosome 15. You have the maternal chromosome 15 on the left and the paternal chromosome 15 on the right. They have all the same genes, all the same loci, but they have different alleles, different imprinting patterns, right? Such that on the maternal chromosome, the Prader-Willi gene is imprinted and the Angelman gene is active. Okay, that means the Angelman gene is producing a protein. The Prader-Willi gene on the mom's chromosome is not doing anything. It's imprinted, it's methylated, it's turned off. Okay? On the paternal chromosome, you have an active Prader-Willi gene and an imprinted Angelman gene, so it's just the reverse. You have the Angelman gene completely turned off on the paternal side and the Prader-Willi gene active, making a protein. Now, as long as you have both of these chromosomes working together, you get the Prader-Willi protein, you get the Angelman protein, and as long as you're making both of those proteins, you won't get the disease. Okay? Now, let's throw in the deletion. Let's go to the left and let's say that the imprinted Prader-Willi gene and the active Angelman gene are deleted from the maternal chromosome. Okay? Now what do we have left? Well, on the paternal side, we still have both of those genes, but the Prader-Willi gene is active, the Angelman gene is inactive. Now, we have enough protein coming from that Prader-Willi gene, that active Prader-Willi gene, so that the child won't develop Prader-Willi syndrome. But remember that the Angelman gene is imprinted. It's inactive. It's not making anything. So the Angelman gene that was active on the mom's side got deleted. The imprinted gene on the paternal side is not doing anything. We have a loss of that protein, and thus those children develop Angelman syndrome. Okay, loss of protein, loss of function, Angelman syndrome. Now, go look at the other side down to the right. Let's say instead of deleting those genes from the maternal chromosome, we delete them from the paternal chromosome. If we do that, what do we have left? We have an imprinted Prader-Willi and an active Angelman gene. That active Angelman gene makes sure it's making the protein that child won't develop Angelman syndrome, but the Prader-Willi gene is imprinted on the maternal side. So we lost the active allele from the dad, and the allele from the mom is shut off. So we're not doing anything as far as that Prader-Willi protein is concerned. The loss of that protein, the lack of function, those kids will develop Prader-Willi. So that's how the gene imprinting will make a difference as to whether the deletion causes Angelman syndrome or Prader-Willi syndrome. That concludes this portion of the lecture. In the next section, we will discuss metabolic pathways and storage diseases.